When we get sick, we often blame viruses and bacteria. Fungi are often overlooked despite causing millions of life-threatening infections every year. Among the most deadly fungal pathogens are Candida albicans, Cryptococcus neoformans, and Aspergillus fumigatus, with Candida albicans as the leading cause of fungal infection in humans. It can cause bloodstream infection, with mortality rate approaches 40% even with current treatment options. Two classes of antifungal drugs are routinely used in the clinic. However, resistance to these antifungal drugs arises frequently and is becoming a major clinical problem. The development of new antifungal drugs remain a difficult process, in part due to the fact that many of the drug targets in fungi are also important in humans. So how can we extend the lifespan of the existing antifungal drugs? Well, first we need to understand how fungi respond to this treatment. As you can see on the bottom panel, when fungal cells are under attack by antifungal drugs, they get really stressed out and become sick. Some will die, but others could actually cope with the stress, recover, and become resistant. This is the same problem that you will encounter with Lysol, which says on its label kills 99.9% of viruses and bacteria. It's the 0.1% that survives and causes us headaches later on. So if coping with stress is what's keeping the fungi alive, then could we be strategic and target the fungi's stress coping mechanism to make the existing antifungal drugs more effective? And that is precisely the focus of my PhD. Specifically, I'm interested in a protein called Cas5. I showed that by removing Cas5 from the equation, fungal cells that are initially resistant to antifungal drugs become sensitive and can even survive a low dose of the drug. So what is this Cas5? Well, it's a transcription factor, which behaves more or less like a light switch. When it's in the active state, it turns genes on. When it's in the inactive state, the genes are off. This is important because the genes that are being turned on by Cas5 in face of stress act more or less like emergency responders. So if you imagine fungal cells being bombarded with an arsenal of antifungal drugs as a sinking ship, then these genes function to repair the hull damage and pump out the water. By identifying these genes, I can begin to assess their potential as new drug targets. If the same gene is found in human, then it's not a good drug target because the patient would also suffer from treatment. However, if the gene is unique to fungi and not found in human, then it's a more viable option, and the patient is less likely to experience side effects. So ultimately, to sink the ship, you have to take out its emergency repair crew, and that is how you kill a fungal pathogen. Thank you.